présenterons la médaille, il nous exposera ses travaux, et ce n'est pas fini, puisque nous aurons encore 2 beaux exposés scientifiques en sciences de la vie, avec éditeur et Denis Duboul, je crois que c'est Denis Duboul puis l'éditeur, peu importe. Et ce n'est toujours pas fini, parce qu'après, vous avez le droit à un cocktail pour célébrer tout cela. Voilà, alors je commence par vous présenter notre lauréat de la Grande Médaille. Je commence en vous disant, pour rappeler des choses que vous savez tous, que la cellule est l'unité de la vie, des bactéries à l'homme. Et les cellules ont 3 propriétés essentielles qui les caractérisent. Elles naissent habituellement par division, dans quelques cas pour le zygote par fusion. Elles croissent et elles meurent. Avec Michael, nous allons entendre parler de la croissance des cellules. Donc une des 3 grandes propriétés biologiques des cellules, au niveau de la cellule. Sachez aussi que la diversité de volume de biomasse cellulaire varie de façon phénoménale entre espèces vivantes et elle est souvent sous-estimée. Si je vous interrogeais, je pense que vous ne donneriez pas les bonnes réponses, la plupart d'entre vous, même les biologistes peut-être. C'est-à-dire que même si on ne considère que les organismes pluricellulaires, il y a 5 ordres de grandeur de différence de taille entre les cellules les plus petites, comme le sperme ou les lymphocytes, et les plus grandes, les adipocytes et certains neurones. 5 ordres de grandeur. Donc, il faut imaginer la correspondance en termes de régulation, de croissance et de maintenance des cellules. L'histoire de Michael débute avant Michael, si je puis dire, enfin avant le travail de Michael, il était déjà né. Elle débute en 1965 à l'île de Pâques, Rapa Nui, en langage local des indigènes de cette île, où des chercheurs ont identifié une bactérie qui dispose de propriétés antibiotiques, en fait antifongiques, c'est-à-dire capable de tuer des champignons. Donc il était envisagé de développer cette molécule comme un médicament antifongique. En fait, cette molécule antifongique s'est avérée toxique parce qu'elle est immunosuppressive, donc c'est un peu embêtant de traiter des champignons en supprimant la réponse immunitaire des patients infectés, mais l'immunosuppression est en soi intéressante. Elle a été nommée rapamycin, évidemment en rappelant Rapa Nui, de l'île de Pâques. Et cette activité immunosuppressive est liée à une inhibition de la division cellulaire. Mais jusqu'aux années 80, 1980, le mécanisme d'action de cette molécule, la rapamycin, qui est devenu un médicament, entre autres immunosuppresseur, restait largement incompris. Comme l'étaient d'ailleurs, d'une manière générale, les mécanismes qui régulent la croissance cellulaire. Mike me rappelait ce matin qu'en fait, à l'époque, on pensait que la croissance cellulaire était un phénomène non contrôlé qui était simplement dépendant des nutriments dont les cellules disposaient, sans qu'il y ait de phénomène très précis de régulation de cette croissance. Et c'est alors qu'intervient Michael Rôle. Il a étudié les mutants de levure, donc petits champignons, qui résistaient à l'action antiproliférative de la rapamycin. Et il a identifié 2 gènes dont les mutations rendent compte de ce phénotype de résistance. Il les a appelés TOR1 et TOR2, TOR signifiant Target of Rapamycin. Cette découverte, en fait, depuis lors, donc ça se passe, il a publié ses premiers articles en 1987, si je me trompe, non, 91, si je ne me trompe pas. Cette découverte a bouleversé la biologie cellulaire et spécifiquement la biologie de la croissance cellulaire. Au passage, notez que ces petits organismes modèles, relativement simples, la levure unicellulaire ou bien un petit ver C. elegans, sont les organismes modèles dont sont venues les grandes avancées fondamentales de la biologie cellulaire autour de la compréhension des mécanismes principaux de la division des cellules, la mort des cellules et donc de la croissance des cellules avec Michael. Et toutes ces découvertes, y compris la sienne, concernant la croissance des cellules, sont de portée universelle, puisque cela a été montré ensuite. Elles ont valeur pour tous les eucaryotes, donc tous les êtres composés de cellules à noyau, de la levure à l'homme. Et donc, ces systèmes ont été conservés au cours de l'évolution des mécanismes mis en jeu. Donc, je reviendrai dans un instant de façon un petit peu plus détaillée sur les travaux de Michael, mais je pense que c'est le temps de m'arrêter pour un instant, vous parler de Michael lui-même. Donc, Mike est né à Porto Rico. Et il a passé une partie de son enfance dans plusieurs pays d'Amérique du Sud, avant ensuite de poursuivre ses études universitaires aux États-Unis, à Chapel Hill. Et son intérêt initial Je crois qu'il voulait d'abord être médecin. C'est assez rapidement estompé. Bon, du coup, à la fois, je trouve que c'est dommage et en même temps, vu le résultat, c'est peut-être pas si mal. Et pour se porter donc de la médecine, il a basculé vers la biologie moléculaire. Il a fait son PhD à Harvard. Puis, il est venu à l'Institut Pasteur avec Maxime Schwartz, ici présent, pour s'imprégner de la grande école Jacob Monod du développement de la génétique et de la biologie moléculaire. Ensuite, il a fait un postdoc à San Francisco avec Ira Hershkovitz dans le domaine de la génétique et la biologie moléculaire bactérienne. Chercheur ensuite indépendant, en 1987, il s'établit à Bâle. Donc voilà un exemple, peut-être on y reviendra, d'un chercheur venant d'Amérique, du Sud et du Nord, et qui s'installe en Europe à Bâle, où il est toujours dans un institut de recherche fondamentale prestigieux, dont il dit qu'il a toutes les qualités, cet institut, pour y mener la meilleure recherche, qui s'appelle le Biozentrum, qui est associé à l'Université de Bâle. Donc, depuis 1987 jusqu'à aujourd'hui, il dirige un groupe de recherche, et c'est là que l'aventure TOR, Target of Rapamycin, s'est déroulée. La reconnaissance de l'importance de ces découvertes lui ont valu, après un certain temps de latence, de reconnaissance effective de l'importance de ce travail, une notoriété, donc il lui a valu néanmoins une notoriété dont témoigne l'obtention d'un très grand nombre de prix scientifiques. J'en cite juste quelques-uns: le prix Louis Jantet, le Breakthrough Prize, le Gerner Canadian Prize, le prix Lasker ou encore le prix Sjöberg. Il est évidemment membre de nombreuses académies, dont la National Academy of Science. Et je voudrais signaler au passage que son intérêt pour la promotion et la défense de la recherche scientifique l'ont amené à prendre des fonctions à un moment à la tête de l'EMBO, le European Molecular Biology Organization. Alors pourquoi tous ces honneurs? Si je reviens maintenant à l'objet de son travail autour de ces molécules TOR. Donc en 91, il découvre TOR 1 et TOR 2 chez la levure. Ensuite, d'autres équipes, mais aussi la sienne, découvrent les équivalents chez les mammifères. Et Michael montre à la fin des années 90 que ces molécules exercent donc un rôle central dans le contrôle de la croissance cellulaire, je l'ai déjà dit, développement de l'anabolisme et régulation négative du catabolisme cellulaire, donc phénomène universel chez tous les eucaryotes. Ces découvertes, en fait, sont conceptuellement importantes parce qu'elles dissocient la notion de régulation moléculaire précise de la croissance cellulaire de celle de la division cellulaire. Jusque-là, les deux concepts étaient mêlés, et parfois on parlait de croissance cellulaire pour parler de division cellulaire. Ici, on parle bien de la croissance, de l'augmentation de volume, de la biomasse. Je suis sûr que Mike va nous l'expliquer très bien dans un instant. Donc conceptuellement, c'est aussi une avancée majeure. Donc quelques éléments, quelques contributions exprimées de façon un peu plus précise. Donc son laboratoire va identifier des complexes protéines. Ces molécules TOR 1 et TOR 2 sont associées à toute une série de protéines. Ce sont des complexes qu'on appelle maintenant mTOR, comme moi chez les mammifères. mTOR 1 et 2, c'est le C ajouté à TOR pour complexe, numéro 1 et numéro 2. Et les molécules TOR sont des enzymes qui catalysent la phosphorylation, qui ajoutent des radicaux phosphore à des acides aminés précis que sont des sérines et des thréonines. Donc, les spécialistes sont des sérines, thréonines, kinases. Et ensuite, Michael a identifié de très nombreux substrats de ces deux enzymes. Et juste, je ne sais pas si c'est vraiment aujourd'hui encore plus complexe, mais dans une superbe revue sur le sujet qu'il a écrite il y a 2 ans, il mentionnait que le complexe TORC 1 avait au moins 56 substrats et TORC 2 26. Donc juste pour vous donner une complexité ensuite des phénomènes seconds qui sont induits par l'activation de ces kinases. Et qu'est-ce qu'il en résulte? Toute une série d'événements de synthèse favorable à la croissance cellulaire, synthèse de protéines et d'autres constituants, lipides, etc. Il a aussi mis en évidence que les deux complexes 1 et 2, TORC 1 et TORC 2, ont des fonctions distinctes et que ces fonctions sont distinctes malgré le fait que les sites catalytiques des deux protéines sont très proches, mais donc que ce sont les autres éléments de la structure de ces protéines qui définissent les fonctions différentes, et il l'a fait par des analyses fines de structure macromoléculaire de ces complexes. Enfin, il a identifié et largement contribué à identifier les ligands, les nutriments, l'insuline, etc., qui induisent l'activation de TORC 1 et TORC 2, et donc des substrats et des voies, des multiples voies, et complexité des voies induites dans l'activation de ces enzymes et leur régulation. Bon, bref, globalement, une contribution, je pense qu'on peut considérer, que nous avons collectivement considérée comme décisive à la description et la compréhension de phénomènes d'une très grande complexité, mais majeurs, encore une fois, dans la biologie des cellules. Travaux qui ont été menés en utilisant toutes les armes possibles de la biologie, de la génétique à l'ultrastructure, en passant par la biochimie et l'imagerie fine subcellulaire. Donc une œuvre fondamentale, considérable, dont l'impact, par ailleurs, en médecine est majeur, dans les domaines de la physiopathologie de nombreuses maladies, à commencer par le diabète, parce que l'insuline, par exemple, est un agent inducteur de l'activation de ce système TORC, mais aussi des maladies du système nerveux et globalement du vieillissement. Incidemment, la semaine dernière, Michael a reçu un nouveau prix prestigieux, le prix Balzan, du fait de sa contribution de ses travaux à la compréhension, peut-être à l'intervention sur les phénomènes du vieillissement. Et puis enfin, et last but not least, l'intervention dans le domaine du cancer. La plupart des cancers sont associés à une hyperactivation de ce système. D'ailleurs, la rapamycin a été utilisée depuis un certain temps comme agent anticancéreux, et évidemment, c'est une voie de recherche absolument majeure que Michael continue de développer. Je dirais que Michael a donné ses lettres de noblesse, entre guillemets, au métabolisme cellulaire en dévoilant les mécanismes moléculaires par lesquels il s'insère dans les fonctions vitales de nos cellules, en rendant compte de beaucoup d'entre elles à travers leur croissance. Donc, pour toutes ces raisons, c'est un très grand honneur pour l'Académie des sciences de décerner à Michael sa plus haute récompense, la Grande Médaille. Donc, c'est au nom de 2023 et ça se passe en 2024. Récompense attribuée aux meilleurs chercheurs dont les travaux ont contribué, quel qu'en soit le domaine, à l'avancée des connaissances. Je vous rappelle par exemple que les 2 dernières années ont été, avant Mike, ont été ainsi reconnues Catalina Carico pour le vaccin contre SARS-CoV-2 et l'année dernière Terence Tao, un très, très grand mathématicien dont je pense que vous vous souvenez tous. Je voudrais ajouter que Michael est un homme que je considère comme avisé, d'une très grande culture scientifique, un sage avec lequel il est toujours extrêmement plaisant et instructif de converser, et vous en aurez un peu l'occasion à l'occasion du cocktail. Enfin, je voudrais ajouter que le parcours de Michael est relativement atypique, puisque nous avons un chercheur américain qui vient s'installer en Europe, ce qui n'est pas le schéma habituel, comme chacun d'entre nous le sait. Et peut-être que d'une certaine façon, il est par lui-même, par son parcours, un plaidoyer pour défendre la recherche scientifique européenne, ce à quoi il contribue. J'ajoute au passage que Mike est francophone et francophile, et que c'est bien notre chance. Donc Mike, au nom de toute l'Académie des sciences, je souhaite vous féliciter très chaleureusement d'avoir ainsi éclairé notre lanterne scientifique par ces travaux aussi fondamentaux. Vous êtes un des rares à l'avoir accompli. Merci beaucoup.
Chers membres distingués de l'Académie des sciences, chers invités, chers amis, je voudrais tout d'abord dire que je suis très honoré et reconnaissant de recevoir la Grande Médaille de l'Académie des sciences. Je suis également fier et heureux. Fier parce que je pourrais dire à mes petits-enfants que moi aussi, j'ai gagné une médaille à Paris en 2024. Mais surtout, je suis heureux parce que cette médaille intègre 2 choses qui me tiennent au cœur. la science et la France. Je vous remercie. Yes, I thank you very much for this wonderful honor. And what I'd like to do now is tell you the story of TOR, the work that's being recognized today. I will give you a long introduction. I will show you no actual data. And the story of TOR is about a protein which is called TOR, targeted rapamycin. And the function of this protein is to control cell growth, as you heard during the introduction. So let me go back in time. I'll tell you not only the story of TOR, but I'll tell you this from a historical perspective. So let's go back in time. So this is a condensed version of the evolution of the TOR field. Over here on the left, you have the very first ever model of TOR signaling published. It's one we published shortly after our discovery of TOR in 1991. I'm actually a little embarrassed we published this model because there's almost no information of value on here at all, more questions than anything else. It actually reminds me of, in America, what would be a Gary Larson cartoon. I don't know if many of you remember Gary Larson. The story is dramatically different today, and this model is already woefully out of date. We stopped drawing models of TOR signaling on two-dimensional pieces of paper because the complexity was just too much. And what drove this increase in complexity in this field was this exponential increase in publications in this area. I'm told today there are over 7,000 publications per year on TOR. And what's driven this increase in publications in this field is the fact that TOR turned out to be fundamentally important and clinically important, which is, of course, combines two large research communities publishing in this area. But the story didn't start in 1991. It actually started earlier. As Alain alluded to, it started in 1965 when a group of scientists went to Rapa Nui, or Easter Island, to collect a soil sample. And what these scientists were doing was prospecting for exotic microbes which might produce some novel metabolite, which they could then develop into a drug. In this case, they were looking for a novel antifungal. They did indeed find such a molecule which had antifungal activity, and as they started to develop it, they realized it had the undesirable side effect of suppressing the immune system. And it was therefore no longer developed as an antifungal. Years later, as surgeons were starting to develop transplantation surgery as a viable medical procedure, they remembered that there had been this molecule many years before which suppressed the immune system. And that's precisely what these surgeons wanted. They suppressed the immune system so the body would not reject a foreign organ. So then rapamycin was now rediscovered and applied for the very reason in which it originally had been rejected, to suppress the immune system. And if you go, so it had been repurposed from an antifungal to an immunosuppressive drug. And if you go to Rapa Nui, you'll actually find a plaque commemorating this act of picking up a handful of dirt from which this bacterium was isolated to produce this secondary metabolite. This plaque, for reasons I don't understand, is in Portuguese, but it says, "In this place was obtained in January of 1965 samples that not only allowed the isolation of rapamycin, but also inaugurated a new era in transplantation surgery." So this was a major development in medicine, the ability to transplant an organ. Having worked on rapamycin much of my adult life, I always wanted to travel to Rapa Nui to see this plaque, a trip to Mecca of sorts for me. I was finally able to do that, and here we are. When we got there, we dropped our bags off at the airport. The moment we got there, rushed out into the bush to see this site, and much to our disappointment, the plaque had been stolen. So we still haven't seen the plaque. We're still looking for it. Of course, a big disappointment. But now back to rapamycin. This is what rapamycin actually looks like, this natural metabolite secreted by soil bacterium. And now it has applications in 3 therapeutic areas. It was originally developed as an immunosuppressive agent for transplantation surgery, but now it's even more famous as an anti-cancer drug, and it's also now used in the treatment of cardiovascular disease. So there was a lot of interest in this molecule when we started working on it. It was known that it was effective for transplantation surgery at the time, but no one understood the underlying molecular mechanism of action of this molecule. Why does it suppress the immune system? And I had a excellent postdoc in my laboratory at the time, Joe Heitman, who had a medical degree and was interested in understanding this medical aspect of rapamycin. And fortunately, we had a local pharmaceutical company in Basel which could supply us with rapamycin, which was not yet fully developed and was not yet in the market. There was no place actually to get it in the world except this one place, which happened to be in Basel. So we're very fortunate to be in Basel. So what Joe and Rao did was they decided to study the mechanism of action of rapamycin by trying to understand what the target is. What is the target of rapamycin? What protein does rapamycin bind in the cell to elicit these effects of immunosuppression? The experiment and what they did, which was an extremely brilliant idea, I thought, and also risky, was to try to use yeast and yeast genetics in particular to characterize the mechanism of action of a drug that at the time was being developed for use in humans. So many of our colleagues were very surprised that we would use lowly yeast, a unicellular organism, to characterize a drug that was being developed for the clinic and use in humans. It was viewed as completely non-physiological what we were doing. It was analogous to giving aspirin to a yeast cell. Why would you ever do such a thing? But they remembered, Joe and Rao remembered, that rapamycin had originally been isolated as an antifungal. And of course, yeast is a fungus, so they did not view it as a molecule to be given to humans, but as a molecule which evolved to work on other fungi in the microbiological warfare that occurs in the soil for microbes fighting for resources in that ecological niche. And the experiment that Joe did in this case was very simple. He simply made a petri dish with agar in it, in which he'd added some rapamycin, grew up our lab culture, our lab strain of yeast, and plated it on the agar. And if there were no rapamycin on the agar of this petri dish, there would be a complete lawn of yeast cells growing there. You wouldn't see the agar underneath. But because there was rapamycin in the agar, the yeast cells were all killed, except for the few spontaneous rapamycin-resistant mutants which arose. So Joe then picked these individual colonies, individual mutants, purified them, and then characterized them genetically. And what he found was that these mutants acquired rapamycin resistance by mutation in any one of 3 different genes, and these genes were FPR1, TOR1, and TOR2. FPR1 was a gene which we'd already characterized in the lab and therefore already had this name. This gene encodes a protein called FKBP, and in fact most of the mutations we obtained fell into this gene, and all these mutations for the geneticists in the room were recessive. Rarely we obtained mutations in 2 novel genes never seen before, and these mutations were not only rare, but they were dominant. So why did we get mutations in 3 different genes? Why were some common and recessive and others rare and dominant? And the answer to these questions comes from the mechanism of action of rapamycin. So this is how rapamycin acts. So here again we have rapamycin, but now drawn in a space-filling configuration. It's a very lipophilic hydrophobic molecule, so it simply diffuses across the plasma membrane to get into the cell. It doesn't require any transporter, which could be mutated to confer resistance. And once in the cell, it binds a small protein called FKBP, which is the product of the FPR1 gene. and it is then this FKBP rapamycin complex which is the toxic agent. Rapamycin alone has no effect. It requires binding to FKBP first. And why that is, I'll explain later. But FKBP is not essential for the cell. It can be deleted in the cell, is fine. And so this is why, although it's essential for drug action. So this is why any simple loss of function mutation in the gene encoding this protein leads to complete rapamycin resistance. And this is why most of the mutations were in this gene and why they were recessive. This, on the other hand, is TOR. TOR is a very large protein. This is a 14 kilodalton protein. TOR itself is about 300 kilodalton. So this is just a very small part of the TOR protein. This is the so-called FKBP rapamycin binding domain in the TOR protein. And all the mutations, and unlike FKBP, TOR is absolutely essential for viability. If you knock out the TOR gene and the TOR protein, the cell cannot survive. So the TOR protein cannot tolerate any very much mutation, or the cell would die. And so every mutation we obtained, which fell in the TOR gene, all of these mutations fell in the exact same codon, which specified a residue in this alpha helix, which is a key binding site for rapamycin. And this is why these mutations were so rare, because they're confined to a single codon within a very large gene, which encodes the large TOR protein. And this also explains why these mutations were dominant. Now, so these mutations were extremely informative. They elucidated the mechanism of action of rapamycin, the fact that it has to form a complex with FKBP first. They define the rapamycin binding site within the TOR protein, but most importantly, they led to the identification of the TOR protein itself, which we identified by then cloning the mutated genes from these yeast mutants. Once we cloned the genes for the TOR genes and sequenced them, we found, first of all, the two TOR genes encoded two very homologous proteins, very similar proteins, and they turned out to be the founding members of this novel class of atypical protein kinases called PIKs, PI kinase-related protein kinases. This means that they all look like defined lipid kinases, but in fact, all members of the class are protein kinases, and to this day, we don't understand why they look like lipid kinases, but are in fact protein kinases, and none has ever been shown actually to be a lipid kinase. Since our early discovery of TOR in yeast, taking this genetic approach, TOR has been now found in all eukaryotes, from yeast all the way to human, including plants, worms, insects, flies, all the way to human. And the nomenclature was to keep the TOR name, target of rapamycin, first given in yeast, with a prefix to indicate the organism from which it came. So mTOR stands for mammalian TOR, but now it's also been used to refer to it as mechanistic TOR. So this is where we were in about the mid-1990s. We had what was a highly conserved essential kinase, which is the target of rapamycin. We elucidated the mechanism of action of this medically important drug, but we still did not know what the function of TOR was in the cell. What physiological role does TOR have in the cell? It did not evolve simply to be the target of rapamycin. What we discovered, and this was a very lengthy period in the lab, which I will not go through, but what we discovered is that the role of TOR is to control cell growth. And that's most graphically illustrated in experiments not done by us, but experiments done in those who worked in Drosophila. And here's experiments done by two Drosophila geneticists, Tom Neufeld and Bruce Edgar, both in the United States. And this here is what's called the fat body of the fruit fly, which is equivalent to the liver of a fruit fly. And what Tom Neufeld did here was he selectively inactivated the TOR protein in these two green cells. And as you can see, these cells ended up being much smaller than the neighboring wild-type cells. Bruce Edgar did the inverse experiment. He hyperactivated TOR, again, in the green cells. The green was the trick to know in which cells he'd actually inactivate or activated. And as you can see, this is the same tissue. These cells are the same size for scale. In this case, when you hyperactivate TOR signaling, the cells get much larger. So the obvious conclusion from this is the role of TOR is to control cell growth, increase in cell mass, cell size. Now, Tom Neufeld took this experiment one step further in isolating a TOR mutant fly. Now, all the cells of the entire fly are defective for TOR signaling. It's important to point out this is what's called a partial loss of function, because if this had been a complete loss of function, I already told you this is an essential protein, this fly never would have been born. So this fly has about 20% normal TOR levels. And as you can see, Tom Neufeld obtained this smaller but perfectly proportioned fly. And then he asked the question, why is this fly smaller? Is it smaller because it has fewer cells, all of normal size, or does it have the normal number of cells, but now individual cells are smaller? Is it a case of cell number or cell size? And the way you can answer that question is by looking in the wing, which is a plane of cells. So you can easily count the circumference of cells, and you can count also the number of cells, and from that you then extrapolate to the rest of the cell. And what Tom Neufeld found was this fly has more or less the normal number of cells, but now each individual cell is smaller to make this smaller but perfectly proportioned fly. To my mind, this is an absolutely astounding result, because to make a fly like this is a program in 4 dimensions, and here this program has been rewritten, at least in the 2 dimensions we can see here, by a single mutation in the genome, which brings down the level of the TOR protein. You can think of all the regulatory circuits, metabolic circuits, which have to be rewired to get this smaller but perfectly proportioned fly. It's also important to realize TOR controls growth in many other physiological contexts, oftentimes independent of development, as would have been in this fly. And here's some of my favorite examples. In the liver, if you fast or feed, your liver changes in size. This is not because you acquire more liver cells or lose liver cells, but your existing liver cells change in size. Muscle, a very common example. If you exercise, your muscles get bigger. Again, you don't acquire more muscle cells, but your existing muscle cells simply get bigger or smaller if you don't exercise. In the central nervous system, there are very dramatic changes, not only in cell size and cell shape, and this has all been shown to be controlled by TOR signaling. And even someone has shown that what I call in nature, queen bees are larger. There's the larvae which give rise to a queen bee have been fed a super nutritious nutrients, so-called royal jelly, which upregulates the TOR protein in the larvae, which then give rise to the bee. So the take-home lesson here is, the take-home message is that TOR controls cell growth very broadly in terms of physiological context and also broadly in terms of organism. Essentially, any case in eukaryotic biology where you see change in a cell size, there's a high likelihood that TOR is mediating this change in growth, cell growth. So how does TOR do all this? How does TOR control cell growth and cell size? Well, work from a large number of labs, including our own, has shown that TOR controls many cellular processes which collectively determine mass accumulation. And these processes can be subdivided into 2 groups: the anabolic processes, which TOR activates, and the catabolic processes, which TOR inhibits. So what TOR does is it balances these opposing forces of synthesis and degradation such that the cell accumulates the appropriate amount of mass relative to, and this is important, relative to nutrients. So TOR is activated by nutrients. So in very simple terms, TOR is what makes us grow when we eat. So how does TOR actually control all these cellular processes? Well, there are effector pathways via which TOR, which emanate from TOR and intersect with key proteins involved in all these processes. In some cases, we know what these pathways are, these biochemical pathways are. In some cases, we don't. But this is where we were in the year 2000. And again, this is a yeast cell. And we've been taking many genetic approaches to further define the so-called TOR signaling pathways, the biochemical pathways via which TOR controls all these processes. And the picture that emerges is that There are, in fact, two branches of TOR pathways. This pathway over here is we call the TOR2 unique pathway because only the TOR2 protein can function here. And this controls organization of the actin cytoskeleton, which determines where new mass is laid down in the cell. So we view this pathway as mediating spatial control of cell growth. This branch over here, which is itself made up of several pathways, controls these processes which lead to mass accumulation in response to nutrients. And so we view this branch over here as mediating temporal control of cell growth. And of course, cell growth has to be controlled both in time and space, and these have to be integrated, these two parameters of cell growth. And what better way to do that than to have the same proteins doing both? So we think that's the logic of this overall TOR signaling network. But at this stage, we still had many questions. For example, what determines the specificity of TOR signaling? Why can either TOR signal through here, but only TOR2 signal through here? We also learned that only TOR over here is sensitive to rapamycin, yet TOR2 over here is resistant to rapamycin, naturally resistant to rapamycin. So what's the difference between TOR here and TOR2 over here? So to answer these questions, we then decided to take a biochemical approach. Everything we've done until now had been genetic. And what we did was we purified the TOR proteins directly out of yeast cells, but using a very gentle purification scheme so that if there were any interacting proteins, we would co-purify them. And once we did that, we would know what proteins TOR interacted with. We could understand how it functions, how it was regulated, and how it in turn controlled other processes. We did that. We purified two TOR complexes, which we call TORC1 and TORC2. And once we had these two complexes purified in a test tube, we could sequence the protein. With the information we got from sequencing the protein, we could then isolate the gene. We isolate the gene, we could then knock that out. And we did that. We found that these two complexes corresponded to these two previously identified branches of TOR signaling. So this was a major breakthrough in the field because it answered many things. It explained the specificity of TOR, why either TOR can signal through here. And the answer is, of course, that either TOR can assemble into this complex, but only TOR2 can assemble into this complex. We also learned why this TOR is rapamycin insensitive. It's sensitive here, but insensitive here. And the answer is, as I'll show you later, that one of the subunits here masks the rapamycin binding site in TOR here, whereas that rapamycin binding site is exposed here. So this is also a breakthrough because it told us, in fact, TOR is two structurally and functionally distinct kinases signaling through these two different branches. So this is where we were in 2002. We then wondered whether what we discovered in yeast was conserved all the way to human. I remember TOR itself is conserved all the way from yeast to human. Are these complexes conserved? We looked in genome databases at this time. There was no complete mammalian genome sequenced yet at this stage. There was a complete yeast, but not a mammalian genome. And so these are the proteins which we've identified as being components of these two TOR complexes. And we found some genes in the incomplete mammalian genome sequence that had some similarity, which suggested that these complexes might be conserved all the way from yeast to human. So we did the actual experiments. We cloned these genes from mammalian cells and found that indeed the two complexes are conserved all the way to human. The same proteins which form these complexes in the yeast form them, or at least their orthologs, form them in mammalian cells. So the picture that's emerged is that this is the TOR signaling pathway is an ancestral or primordial signaling pathway conserved all the way from yeast to human to mediate this very fundamental process of cell growth. Now, this statement I just made is not entirely accurate because this part of the signaling pathway up here, the signaling network up here, is the insulin or growth factor signaling pathway, and this evolved much later. This evolved with multicellularity, and then it was grafted onto the more primordial TOR signaling components down here. And the reason for that is that growth control in multicellular organisms is more complex than growth control in a unicellular yeast, because in multicellular organisms, it's critical to control the growth of every cell in the body relative to every other cell in the body, such that our organs end up being properly proportioned. That then is what signaling through the growth factor signaling pathway provides, which controls growth over a whole body plan. And now we know this is combined with local nutrients and local energy in the form of ATP. Once these three factors are present, growth factor, nutrients, and energy, these are all the conditions which allow growth. The TOR pathway is activated and growth ensues. Now, another fascinating aspect of the TOR signaling pathway is the unusually large number of diseases to which it's been functionally linked. These are all diseases which arise from mutations in these components of the TOR signaling pathway. And what all these diseases have in common is they're characterized by inappropriate cell growth. Again, underscoring the concept or the notion that TOR is a central controller of cell growth. And this can be malignant forms of cell growth or benign forms of cell growth, but in all cases, excessive cell growth. More interestingly, TOR has been implicated in another set of disorders, the metabolic disorders. We know that chronically high circulating levels of nutrients can activate TOR even in the absence of growth factors, and this leads to obesity, adipogenesis and obesity, which can in turn lead to type 2 diabetes. There's a more direct link to diabetes by this negative feedback loop where TOR phosphorylates and inhibits this protein, the insulin signaling pathway, which results in insulin insensitivity. In this case, rapamycin has actually been proposed for treatment as an anti-diabetic, because if you inhibit TOR, you inhibit the negative feedback loop, and you restore signaling to the insulin pathway, restore insulin sensitivity. I don't think any pharma company is developing rapamycin as an anti-diabetic, because there are a number of complications which would prevent that. But something giving some credence to the notion that you might be able to do this is that the world's most commonly prescribed anti-diabetic, metformin, acts at least in part by inhibiting TOR, like rapamycin. Okay, another fascinating aspect of the TOR signaling network is the fact that it extends lifespan. So we've shown in 2009 that rapamycin can extend lifespan in mammals. This is the first pharmacological extension of lifespan in mammals. Rapamycin had already been shown to do this in yeast, worms, and flies, and this paper then in mammals. And this has created a great deal of excitement in the aging field, because it's been known since 1935 that so-called dietary restriction, or eating less, extends lifespan. But nobody understood the molecular basis of this phenomenon. Now, we know that TOR is a nutrient sensor, and the effect of rapamycin is, by inhibiting TOR, is to mimic the state of caloric restriction. The cells cannot sense there are nutrients available, and they don't respond to those nutrients. So this then provides the long-missing mechanism by which caloric restriction or dietary restriction extends lifespan. You eat less, you have less TOR activity, and all the consequential metabolic changes as a result have the beneficial effect of extending lifespan. Now, by this stage, we had a rather good understanding of how TOR controls growth of cells, particularly the cell in which that TOR molecule resides. But here is a very exciting experiment, which was published by Pierre Leopold, a colleague of many of you. He's at the Institut Curie here in Paris. He was a fly geneticist, like Tom Neufeld and Bruce Edgar. And what he was doing, he was looking for flies, mutant flies, which had a growth defect. And he isolated a fly which had a defect in one tissue, the same tissue I showed you before the fly, the fat body. And this mutation affected an amino acid transporter in these cells. So amino acids didn't get in these cells, and therefore the amino acids could not activate TOR. That's the nutrient that activates TOR. As a result, TOR was not active in the cells of this one tissue, and as a result, the cells were smaller in this tissue, and the overall tissue was smaller. But his astounding observation was that in all the other tissues of the body shown schematically here, where TOR should be normal because they all have these cells have their own amino acid transporters, a different amino acid transporter. Despite the fact that these should be growing normally, all the tissues had reduced growth to match the size of the defective fat body over here. So this told us that there is a TOR-dependent signaling between tissues to control growth over a whole body plan, to make sure the organs end up being properly proportioned. And this is then what we wanted to do next. We wanted to study what is this new mechanism of controlling cell growth over a whole body plan, so-called non-autonomous control of cell growth. We didn't want to do this in the fly. We did it in mice. And to do this, we wanted to knock out TORC1 or TORC2 individually in individual tissues. And the way you do that is, I will not tell you how you do that, but to knock out TORC1 or TORC2, we create a situation where we can knock out this Raptor protein, which is unique and specific to this complex, or the Richter protein, which is unique and specific to this complex. So this way we could knock out one or the other. And we could do it in any tissue of choice. We wondered what tissue to study because TOR is, of course, found in all tissues. So we decided to focus on the so-called metabolic tissues because these are the tissues which are most insulin and nutrient sensitive, and they're also the tissues that control energy homeostasis in the body. So these are the tissue, and these are exact same inputs which control TOR signaling. So we thought TOR would have a particularly interesting role in these tissues. Let me just very briefly show you a couple of results we had. This is we've knocked out specifically TOR complex 1 in adipose tissue, so in fat. And this mouse now turned out to be metabolically healthier than a normal mouse. This mouse could eat whatever it wants, a high-fat diet, and never becomes obese like a wild-type mouse shown here. This mouse also has low levels of cholesterol, low levels of insulin, so it's metabolically super healthy, which now harkens back to that paper I showed you before, that rapamycin, which also inhibits growth, inhibits rapamycin, which also inhibits TOR, like the knockout here, extended lifespan. But the experiment I showed you before where they treated mice with rapamycin, it doesn't specify a specific tissue, because when you give a drug, it goes to all the tissues of the body, which begs the question, if there's a specific tissue in which inhibiting TOR creates this beneficial effect of extension of lifespan. And of course here we've inhibited TOR in one specific tissue, adipose tissue, fat tissue, and we get a super healthy mouse. So we think that adipose tissue, perhaps other tissues as well, are particularly important in controlling lifespan. We actually haven't done the experiment to see if this mouse lives longer. Finally, we have another mouse. Now we knocked out TOR complex 2. This mouse had a very different phenotype, different behavior. It became much larger, and this is because it had high levels of insulin and high levels of insulin-like growth factor. And this led us to propose a new axis whereby the cell mediates insulin homeostasis, where adipose tissue signals negatively to the liver and to the pancreas to reduce production of these insulin and IGF, so that when we mutate this, we get high levels of insulin, which is a growth factor, and you get this increased mouse. We spent 10 years looking for the mechanism whereby some TORC2-dependent factors secreted from adipose tissue attacked on liver and beta cells of the pancreas. This failed all the time until an observation we made only a couple years ago, about which we're extremely excited and we're going to continue to pursue, and that is, in fact, there is no secreted factor. Because when we look now in the adipose tissue and look specifically in the nervous system in this adipose tissue, and that's shown here. So here is adipose tissue of a normal mouse, and what we're staining here is for different kinds of neurons, sympathetic neurons, which are neurons which signal from the brain to the fat in this case, or in this magenta, we're staining what are called sensory neurons, which are the ones that signal the opposite way, from the fat to the brain. The sympathetic nervous system is extremely well characterized in fat. There's a large literature on this, but no one had ever seen a sensory nervous system in fat before. So we've discovered a sensory nervous neuronal network which had never been seen before. And the interesting observations, and now we look in the TORC2 knockout mouse, which has these defects outside the adipose tissue, we find that the nerves which innervate the fat, if we look at the sympathetic nervous system, is perfectly normal, but the magenta neurons are missing. Whereas, so we're doing whereas they're absolutely normal over here. Okay, so we've discovered a new neuronal system in adipose tissue, and so we think the way adipose tissue, and we've discovered a sensory nervous system. So we think the way fat is now signaling to the rest of the body is via the central nervous system. And this now presents a completely new picture of how fat induces diabetes. It's not through secretion of factors, but affecting the central nervous system. And this is the model we have to now investigate further. How much time do I have, by the way? Am I? Okay. While we were trying to understand mice in which we've knocked out the TOR signaling, we also want to understand what was the effect of having too much TOR signaling. And to this, we did another approach. We now knocked out proteins upstream of the TOR complexes, which are negative regulators, also known as tumor suppressors. We knock out TSC. We get hyperactivation of TORC1. We knock out this other one, P10, and we get further activation of TORC2, higher kinase activity. But in this case, we also activate TORC2. So, and we did this specifically now in the liver. So what happens when we hyperactivate TOR in the liver? This is shown here. This is the mice now 20 weeks of age. They have very severe liver cancer. Every one of these nodules is a tumor. And we know these tumors are due to hyperactive TOR signaling, giving rise to inappropriate cell growth due to TOR, because if we treat these mice, such a mouse with rapamycin, which inhibits TOR, we protect the liver from cancer. And of course, we view this as an excellent model system to study liver cancer, and this is also something we've been working on recently. Okay, to end, I'll tell you one last short thing. We have been, as I've told you, we've been very focused on defining the physiology and pathophysiology of TOR signaling since our early discovery of TOR back in 1991 in yeast. But we've also tried to understand TOR at the atomic level. So we recently determined the structure of TOR complex 1, human TOR complex 1. This is a very large complex. It's the size of a ribosome, much bigger than I expected it to be. It also turned out to be a dimer of heterotrimers. So this blue-gray is the TOR molecule, and there are two of them. There's another subunit of the complex, and then a third subunit up here. Since it's a dimer, a homodimer, it has two catalytic sites, one here, one here. This is what mediates the phosphorylation reaction of this kinase. And then we also determined the structure of TOR bound to rapamycin. Remember, I told you rapamycin alone can bind TORC, but cannot inhibit it in TORC. But cannot inhibit. Rapamycin needs to bind to FKBP first before it can inhibit. And this is something we never understood until we saw the structure, and that's shown here. So this is that small protein I told you about in the very beginning, FKBP. Rapamycin, you don't see it, but it's very small, but it's a glue, which glues FKBP to the lip of the catalytic site, and this acts as a kind of a lid which blocks access to substrates to the catalytic site. Very unusual mechanism of action of a kinase inhibitor, but there you have it. Also explains why if we take away FKBP, now substrates can gain perfect access. Rapamycin alone just simply does not have the mass to block access to the catalytic site. And finally, we recently determined the structure of TOR complex 2. This was to 3.2 angstrom resolution. In this case, we found that TOR, it's also a dimer. TOR itself, two of them per complex, is the same in both. But now in this case, it's decorated with different subunits. And I think the most interesting observation that came out of this was something that had confused us since 2004, and that TOR complex 2 is insensitive to rapamycin. This is what I told you before. Rapamycin can bind inhibit TOR in TOR complex 1, but not in TOR complex 2. It's the same protein. Why can it inhibit one complex but not the other? And the answer is revealed by the structure. So this, so again, here's TOR complex 1. This is FKBP rapamycin binding to the lid of the catalytic site. It turns out this subunit, which is specific to TOR complex 2, masks the rapamycin FKBP binding site such that FKBP rapamycin can never bind to TOR complex 2, cannot bind TOR in TOR complex 2, explaining why TOR complex 2 is insensitive to rapamycin. Okay, I think I've given an overview of the physiology, molecular biology, the structural biology of TOR signaling. I've now come to the end. I cannot thank all the people in the lab over the last 30-some years who have done this work, but these are the people who are currently in the lab and are still busy trying to make discoveries. So thank you very much for your attention, and thank you for the medal once again.
Many thanks, Mike, for this fascinating overview of your work. I think we should take a few minutes for questions. Yes, please. Perhaps could you just introduce yourself?
Thank you, Mike, for this beautiful seminar.
Hello, could you just introduce yourself?
Oh, I'm Stuart Edelstein, one of the foreign members of the Academy, and I just couldn't resist mentioning that I was on the Scientific Advisory Board of the Biocentrum in the 1990s, and we were confronted with a case of question for promotion of a young scientist. And there were some hesitations, but of course no one realized exactly what the long-range impact would be. And fortunately, the vote was positive.
Well, a belated thank you.
As far as the question I have is, it's beautifully described how the different factors interact. But what ultimately is controlling the expression level of the TOR? Are there issues at the level of transcription factors that also come into play? And for example, take a very obvious example. We know that dog species can vary in weight by a factor of 100. Could this be also something involved with TOR?
When it comes to regulation of the TOR genes itself, this is an extremely boring story because there seems to be little to no regulation. TOR seems to be expressed in all cells, and it has to be there, I think, because if you shut it off, it controls protein synthesis and all these other processes which would be required for its own synthesis. So if it was regulated in a way which you could turn it off, the system would not recover from that. So it always needs to be there so that it can act when it's needed. For example, if you starve an organism, TOR activity goes down via its upstream regulation, but the TOR proteins all remain. Okay? They even remain in our cells when we stop growing as adults. But here they take on a different role. This is how they take on control of aging. They drive the aging process. But they're always there.
Thank you.
Nesh.
Mike,
Nesh.
with your liver tumor model, if you expose the mice to caloric restriction or amino acid starvation, did you block tumor formation?
We didn't actually do that experiment. We probably have to deprive them of too many nutrients, and that would in itself have detrimental effect. But there's a long history of people trying to treat cancer via the diet. And this is usually some limitation in the diet, some sort of dietary restriction. So there is reason to believe that eating less could help in some context to a limited extent. But we have not done that ourselves.
3.
je suis physicienne, un peu chimiste et pas du tout biologiste. Hein.
Et vous êtes où
Moi, je suis là.
Ah, d'accord.
Et donc, ma question va être très simple. Vous n'avez pas du tout parlé du noyau des cellules. Est-ce que, quand vous avez montré vos mouches, la petite et la plus grosse, en disant que c'était une homothétie de la taille de la cellule, est-ce que la taille du noyau varie de même?
There is a great deal of work which suggests that the size of the cell is determined by the size of the nucleus. I think that's what you're referring to. We have not looked at that. That experiment I showed you is not from our lab. That's the fly geneticist Tom Neufeld. But my feeling is that the nucleus is probably not changed in size in those cells. Don't ask me why I feel that, but that's we don't know. We don't know why. No. We know that the cells are smaller because there's less TOR signaling, so it makes less proteins, less of everything. So I do not know also what determines what new size these cells adapt. When you reduce TOR activity 20% or the cell is 20% smaller, what determines the threshold of cell size? This is also completely unknown. There is a lower level, yes. It has to be bigger than the nucleus, that's for sure. Yeah.
Antoine?
Michael, I have a naive question, a cell biology question. The volume of the cell depends on the set point between insertion of membrane and retrieval of membrane, in a way.
And the what of membrane?
On the set point between insertion of membrane and retrieval of membrane.
Yeah.
And I guess that metabolism, in a way, regulate the level of that set point. But what is the mechanism?
The TOR controls all these anabolic processes, lipid synthesis that makes the membranes, the proteins that go into the membranes, nucleic acids which make the DNA which goes into the nucleus. But this is, again, goes through a greater problem. How does the cell integrate all these different factors? And this is why I said I was so amazed by this fly, which is smaller but perfectly proportioned. And again, all these, you have to integrate all these different pathways to get at the right homeostasis of all these events. This, we are very far from understanding that. I think we're just scratching the surface. I think we need some more quantitative biology, maybe physicists who will model this in a way which we can then perhaps test experimentally.
Francis, André, and Maxime, short questions.
Yeah, short question, because I think you did not really comment on this. What do we know on the subcellular location of TOR and its dynamics?
Yeah, TOR has been found to be on the surface of the lysosome, which is an organelle which breaks down unneeded protein and other molecules. Why it's there is not entirely clear. There was an early model that proteins would go into the lysosome, they would be degraded, the amino acids would be released, and it's amino acids that activate TOR, so TOR would be right there where the amino acids would be available. But now it turns out that the amino acids which are in the lysosome are not that important for activation of TOR. They're sensed directly in the cytoplasm. So at one point there was a very beautiful answer to your question, or at least a very beautiful answer to the question of why it's on the lysosome, but it's not clear now why it's on the lysosome, but it's there.
Maxime.
An irrelevant question.
Okay.
Do you have to go to Easter Island to find a microorganism producing rapamycin?
I sincerely doubt it. We've never looked elsewhere. There's a curious fact that if you look in worms or plants which have TOR complexes, they seem to be naturally rapamycin resistant, no matter where that worm came from or that plant came from, depending on the plant. So my feeling is these microbes are everywhere in the world, and you don't have to go to Easter Island. But it was a nice place for scientists to go on somebody else's budget. You know, I think.
And Mike, I have a short question. You showed us these very impressive results in terms of TOR-dependent effect of adipose tissue on on neurons or sensitive system. So could it be that there are other Tor-dependent interactions between cell tissues and the neural system elsewhere? I'm thinking first of the other fat. No, it doesn't come to my mind. The other name.
Did you say the immune system? Between Tor and the immune system?
No, not the immune system.
The nervous
system and other tissues in a Tor-dependent. I'm thinking of the brown fat, perhaps, and other things.
This is something we have to look at. If you knock out Tor in muscle, you get muscular dystrophy, you get breakdown of neuromuscular junctions. But this is not exactly the same situation. This is a direct physical contact. But it could, everything's possible. We don't know at this stage.
Okay, I think we'll stop there. Thank you very much again, Mike.
Thank you.
Bien, maintenant nous allons entendre Denis Duboul. Denis Duboul est professeur au Collège de France, titulaire de la chaire Évolution du développement des génomes. Il est membre de l'Académie des sciences depuis 2005, a travaillé à Genève et à l'école de l'EPFL de Lausanne, maintenant au Collège de France. Donc ses études dans le domaine du développement et de l'organisation des membres font école et sont des sujets de grand intérêt en biologie. Donc aujourd'hui, il va nous dire qu'est-ce qu'il en est de l'amour impossible entre Evo et Devo. Évolution et développement pour les non-initiés.
Merci, Monsieur le Président. Merci aussi de m'avoir convié à cet événement qui me donne l'occasion de féliciter mon cher collègue et ami Mike Rôle pour cette nouvelle distinction. Oui, j'aimerais profiter de cette cérémonie en fait pour vous parler un peu d'une nouvelle discipline des sciences de la vie, discipline récente, qu'on appelle communément l'Evo-Devo, et qui se trouve à l'interface entre d'une part l'étude du développement et celle de l'évolution. Ce sont là deux disciplines qui sont vraiment des disciplines majeures des sciences de la vie et qui, tout en étant intimement liées, sont de nature assez différente, et c'est de cela que j'aimerais vous entretenir aujourd'hui. Alors si l'on commence par considérer la question vraiment fondamentale que pose la biologie du développement, c'est une question qui en fait a trois piliers. D'abord la question de la multiplicité et de la taille des cellules, de leur nombre. Ensuite une question de la diversité morphologique et fonctionnelle de ces cellules, c'est la grande question de ce que l'on appelle la différenciation cellulaire. Et puis bien entendu l'intégration de ces deux paramètres dans un espace à 4 dimensions pour former un système cohérent que l'on appellera un organisme. Et cette question, en fait, est celle de notre ontogenèse, donc de notre origine en tant que personne. De façon assez intéressante, ces trois piliers se retrouvent dans la question de notre phylogenèse, donc de notre origine en tant qu'espèce. D'abord, il est question du nombre et de la taille des espèces, de leur diversité anatomique et fonctionnelle, et puis bien entendu de leur intégration dans un ensemble, un écosystème ouvert, censé être dans un équilibre proche de ce que l'on pourrait appeler une homéostasie du vivant. Alors on voit donc que les termes de ces deux questions sont comparables, et en fait même les représentations graphiques que l'on utilise généralement pour illustrer ces disciplines sont relativement semblables. Comme on le voit ici sur la gauche, vous avez le travail tout à fait extraordinaire d'Edward Conklin en 1905 qui vous montre les premiers branchements cellulaires du développement des tuniciers avec cette structure en branchement binaire avec des ancêtres, avec des extinctions, etc. Et puis sur la droite, vous voyez une phylogénie classique avec les mêmes structures de branchement binaire, avec des questions sur la nature de la variation, sur les points de branchement, du maintien des tissus, etc. Et même aujourd'hui, où les outils que l'on utilise pour ces deux types de classification sont beaucoup plus évolués, on voit que ces iconographies sont toujours relativement semblables. Et vous voyez sur la gauche une façon extrêmement moderne de représenter le développement de la mouche, de la Drosophile melanogaster. Je vous passe les détails. Sachez simplement que chaque point est une cellule, et puis qu'en bas, vous avez le départ du développement, et vous avez cette extraordinaire explosion en différents tissus, en différents types cellulaires. Et puis sur la droite, vous avez une nouvelle classification de l'arbre du vivant, fait avec des outils un peu plus performants. Et vous voyez que lorsque l'on regarde ces deux iconographies de loin, eh bien, on a cet aspect très semblable. D'ailleurs, à la fin du XIXe siècle, ces deux disciplines n'en faisaient qu'une. On pouvait utiliser pratiquement indifféremment le mot de développement et d'évolution pour qualifier chacune de ces deux disciplines, jusqu'au développement de la génétique formelle et surtout de l'embryologie causale, donc l'école allemande en particulier, qui va s'atteler à comprendre les mécanismes en jeu au travers d'approches expérimentales au détriment de leurs origines évolutives. Et ce schisme sera accentué ensuite par l'émergence de la génétique des populations, d'un côté, conduisant à la nouvelle synthèse, alors que les expérimentalistes se scindaient en deux camps, qui sont toujours palpables aujourd'hui, les biologistes du développement au sens strict d'un côté, et puis les biologistes appelés plutôt moléculaires de l'autre, qui vont s'intéresser à la structure et au fonctionnement des gènes. Ces deux disciplines fusionneront dans les années 80-90 pour se refermer, pour donner le contexte actuel dans lequel l'étude globale des mécanismes développementaux est presque systématiquement associée à un aspect comparatif, donc intégrant une pensée évolutive. Et le vrai catalyseur de ce mouvement de rapprochement, c'est la révolution qui va se passer entre les années 1985-1995, qui précisément représente l'émergence de cette discipline de l'Evo-Devo, dont le champ n'est pas très bien défini, dont d'ailleurs la définition elle-même est assez différente selon que l'on vienne de la branche du développement ou de celle de l'évolution. Cette révolution des années 80 se caractérise par l'énoncé de trois grands principes qui sont formulés successivement sur une dizaine d'années. Premièrement, l'universalité des gènes. Ensuite, l'universalité des principes qui sont en jeu pendant le développement. Et puis finalement, l'universalité des génomes, qui est un point qui est peut-être intéressant à discuter. Et cela va conduire à un changement de paradigme total à la fin des années 90, que j'ai illustré ici avec une question: comment les éléphants produisent-ils leurs trompes? Une question que, quand je faisais mes études à l'Université de Genève, on donnait la réponse grâce au gène de la trompe. Or, aujourd'hui, cette question est totalement inversée, puisque l'on sait que l'éléphant n'a pas de gène de la trompe. Et donc, la question que nous nous posons, c'est pourquoi nous n'avons-nous pas de trompe, puisque nous partageons nos gènes avec ceux des éléphants. L'universalité des gènes, c'est une évidence aujourd'hui, bien que l'on essaye souvent, un peu désespérément, de trouver quelques gènes pouvant justifier des traits spécifiques à certaines espèces, la nôtre en particulier. L'universalité des principes du développement est également bien assimilée, tels que par exemple les divisions mitotiques, asymétriques ou la détermination des structures le long de l'axe corporel antéro-postérieur. L'universalité des génomes, la façon dont ils sont structurés, leur organisation fonctionnelle, leurs variations épigénétiques sont également patents aujourd'hui. Et je vous montre là une carte de synthénie, de comparaison des domaines de synthénie, qui montre la réorganisation des chromosomes. À gauche, vous avez le génome humain et à droite, le génome de la souris. C'est une carte des synthénies, c'est-à-dire les couleurs vous montrent les endroits où des grandes parties de chromosomes ont été conservées. On voit des morceaux de 30, 40, 50 mégabases conservés entre ces espèces. Par exemple, au chromosome 2, le deuxième depuis la gauche, vous voyez que le bas est fait de deux couleurs qui correspondent au chromosome 1 et au chromosome 2 de la souris sur la droite. Dans le séquençage de ces génomes, commencera avec des invertébrés, bien sûr, qui tous montreront dans les années 90 un nombre de gènes légèrement inférieur ou autour de 15 000. Et à l'époque, peut-être certains d'entre vous s'en souviennent, on estimait le nombre de gènes chez les humains, à cause de ce nombre de 15 000 chez la mouche, de 120 voire 130 000. Et pourtant, le couperet tombe avec ce nombre d'à peu près 22 000 gènes, qui est notre nombre actuel, rajoutant ainsi une nouvelle frustration à celle émanant déjà de la théorie de l'évolution. Il n'en reste pas moins que les êtres humains, comme les autres vertébrés, ont un niveau de complexité très supérieur à plusieurs égards à celui des nématodes auquel Alain Fischer faisait allusion, ces petits Cenorhabditis elegans qui n'ont guère plus de 950 cellules, un peu plus de 300 neurones, alors que leur nombre total de gènes n'est pas si différent. Et comment cela est-il possible? Eh bien, plusieurs facteurs peuvent expliquer cet état de fait. Premièrement, la complexification des structures géniques, le fait que pour chaque unité de transcription, plusieurs types d'ARN vont être produits, plusieurs types de protéines, des isoformes vont être produites. Ensuite, une diversification des fonctions géniques, un gène qui va être recruté pour faire d'autres choses, dans un autre contexte, à un autre moment du développement. C'est ce qu'on appelle la pléiotropie ou la multifonctionnalité. Cette idée d'une évolution par modification des régulations géniques, plutôt que des gènes eux-mêmes, est une des émanations de l'Evo-Devo, bien qu'elle fut proposée une dizaine d'années plus tôt, d'abord par Marie-Claire King et Alan Wilson, en comparant certaines protéines chez les humains et les chimpanzés, et puis bien entendu par François Jacob dans sa conférence de 1977 à Berkeley sur le bricolage de l'évolution. Cet empilement successif de fonctions, ce recrutement de gènes des développements particuliers pour accompagner l'émergence de nouvelles fonctions ou de structures, peut être simplement contemplé en fait par la simple observation des domaines d'expression de ces gènes, puisque les domaines n'ont pas la même histoire évolutive. Et je vous montre un exemple ici, peu importe de quel gène, de quel groupe de gènes il s'agit, ce n'est pas tellement important, mais vous voyez que c'est un gène qui est exprimé sur la face dorsale de l'embryon à droite, dans le système nerveux central. C'est un gène qui est aussi exprimé au bout des pattes, dans ce qui deviendra les doigts. C'est un gène qui est également exprimé dans les follicules des moustaches. Or, on sait que le système nerveux central est une structure extrêmement ancienne, qui remonte à plus de 500 millions d'années. On trouve déjà chez les procordés. Par contre, les mains et les pieds sont des structures qui apparaissent au Dévonien, donc à peu près à moins de 380 millions d'années, alors que les follicules des moustaches sont des structures qu'on appelle pléiomorphiques, c'est des structures typiques que l'on trouve chez les mammifères, autrement dit à moins 180 millions d'années. Ce sont des synapomorphies en fait des mammifères. Et donc on peut reconstruire une sorte d'archéologie des régulations. On voit qu'un certain gène était d'abord actif dans le système nerveux central, et puis lors de l'émergence des doigts, l'évolution en quelque sorte est allée chercher la fonction de ces gènes pour aider à faire des doigts, puis plus tard pour aider à faire des follicules de moustache. Et puis le troisième facteur important de complexification qui tient aux interactions entre gènes par des réseaux de régulation génétique qui interagissent les uns avec les autres et qui rendent évidemment le système de plus en plus complexe et par conséquent de plus en plus performant, mais également aussi résistant à de nouvelles étapes évolutives. Alors c'est un concept qui dérive vraiment d'un esprit d'ingénierie, celui de Eric Davidson à Caltech. J'ai un peu de peine, je vais m'excuser avec ma machine. Certains de ces réseaux sont plus importants que d'autres. Ce sont ces fameux kernels, par analogie au noyau des systèmes d'exploitation en informatique, que l'on va retrouver chez tous les animaux, car étant indispensables à leur développement. Vous voyez, j'ai projeté sur mon ordinateur une image différente de celle. Je suis absolument navré. Ah, vous l'avez peut-être vu. Voilà. Il s'agit de ces kernels, où vous voyez en fait ici un réseau dont le but est de produire un type de tissu qu'on appelle de l'endomésoderme. Vous avez en différentes couleurs différents types de réseaux de régulation génétique qui vont interagir. Et l'augmentation de ces interactions peut complexifier les systèmes. Voilà. Alors que l'ensemble de ces facteurs contribuent à augmenter la complexité d'un système, en gardant le nombre de gènes relativement constant, ils contribuent également à la mise sous contrainte progressive de ces systèmes, et par conséquent à un espace de variation qui va probablement se restreindre de plus en plus. C'est un point central à considérer quand on pense l'évolution, en particulier lorsque l'on s'intéresse à la nature de la variation, qui, comme vous le savez, est un sujet longtemps ignoré en évolution, en particulier dans l'ouvrage de Darwin, et qui est intimement lié au développement embryonnaire, un développement embryonnaire également négligé comme source de variation au XIXe siècle. Car en effet, c'est bien là le contexte de l'Evo-Devo, qui est que l'évolution se matérialise en partie pendant le développement, et que les mécanismes en jeu lors du développement embryonnaire sont en fait une source de variation évolutive. Mais je pense qu'il est important de bien comprendre que le développement n'explique pas, ne peut pas expliquer l'évolution. Les mécanismes du développement suggèrent simplement des mécanismes évolutifs possibles et peuvent indiquer comment la variation a pu s'y appliquer. Je vous montre ici un exemple qui concerne une des questions vraiment emblématiques de l'Evo-Devo depuis une trentaine d'années, qui est celle de la transition de la nageoire aux membres tétrapodes, une transition donc qui se passe au Dévonien il y a à peu près 370 millions d'années. Et en effet, c'est la compréhension des détails de l'apparition des doigts pendant le développement de l'embryon qui a été essentielle pour proposer des schémas évolutifs de cette transition qui est totalement critique, qui accompagna la sortie des vertébrés du milieu aquatique vers le milieu terrestre. Et par ailleurs, cet exemple illustre très bien les trois piliers théoriques majeurs de l'Evo-Devo, qui sont résumés sur cette image. D'abord, l'existence d'une boîte à outils relativement restreinte, composée de facteurs de transcription, de facteurs paracrines, présents chez tous les animaux et montrant de grandes similarités de séquences. Deuxièmement, ces facteurs de transcription et paracrines sont reliés par les mêmes voies de signalisation, selon des modalités et des principes qui sont bien sûr conservés dans tout le royaume animal. Et puis finalement, ces gènes sont régulés par des enhancers multiples qui ont une grande modularité, de telle sorte que l'expression de ces gènes peut être modifiée de façon locale, intra- ou interspécifique. Ce sont quelques outils simples de construction qui sont présents chez tous les animaux. Le premier de ces piliers, je l'illustre rapidement ici, avec cette image qui vous montre la conservation de certains gènes qui sont en action pour organiser les plans du corps chez les insectes, les invertébrés et chez les vertébrés. Vous voyez dans la partie haute de cette image, les invertébrés, les crevettes, les insectes, et puis dans la partie basse, les vertébrés. Et vous voyez avec ce jeu de couleurs l'expression, les domaines d'expression de certains gènes qui vont spécifier les parties antérieures, les têtes, les parties postérieures. Et vous voyez la grande similarité que l'on trouve entre ces domaines d'expression. Deuxième exemple, sur le deuxième point, on voit ici la voie de signalisation Wnt, qui est une voie très importante pendant le développement précoce, avec de façon très classique un ligand qui se promène entre les cellules, qui va interagir avec des récepteurs sur la membrane, qui va induire un signal qui va passer à travers la membrane avec une chaîne de réaction à l'intérieur du cytoplasme, se terminant par l'entrée d'un signal à l'intérieur du noyau et de l'activation de gènes cibles. Alors ne parlons pas même de la voie TOR qui est à l'honneur aujourd'hui, puisque, comme on l'a entendu, c'est une voie qui est conservée de la levure, où elle fut clonée initialement, jusqu'aux mammifères. Et puis le troisième pilier, c'est l'importance croissante de la régulation génétique et le fait que les gènes du développement, de façon presque systématique, sont régulés par de très grands paysages de régulation, contenant des séquences multiples. Ces paysages sont beaucoup plus grands que les gènes eux-mêmes. Je vous montre ici une taille relative d'un gène en noir, qui sont donc soumis à une très grande modularité dans leur régulation. Et de façon intéressante, on assiste depuis une quinzaine d'années à une transition entre l'idée du gène tout-puissant des années 70, au gène considéré parfois aujourd'hui comme une sorte d'unité exécutive, dépendant de paysages de régulation tout-puissants. Vous voyez ici tous ces interrupteurs en couleur, chacun de ces interrupteurs contrôlant l'expression de ce gène dans un endroit particulier de l'embryon en développement. Laissez-moi terminer en évoquant peut-être deux interrogations associées à cette discipline de l'Evo-Devo. La première concerne le choix des systèmes d'études, car en effet, ceux-ci sont souvent sélectionnés suite à l'existence de traits adaptatifs très marqués, qui peuvent parfois éloigner des principes fondamentaux. Et par exemple, la drosophile, la mouche drosophile, ou les diptères en général, dont on a déjà entendu parler, tout en ayant été une source absolument extraordinaire de matériel à cause de la génétique inégalable de cet animal, se développe d'une façon extrêmement particulière, et c'est pour cette raison-là qu'elle a été choisie. Donc on a cette sorte de dualisme épistémologique où on va choisir un système d'études pour des caractéristiques extrêmement marquées, mais qui vont faire que ce système va s'éloigner en fait de la façon dont se développent non seulement les vertébrés, mais même la plupart des invertébrés. Et on peut donc se demander d'une façon un peu théorique s'il faudra étudier l'ensemble de tous les animaux pour arriver à une compréhension satisfaisante, ou si d'un point de vue épistémologique, il ne vaudrait pas mieux d'étudier simplement le système le moins approprié à la question que l'on se pose. Et puis, une deuxième difficulté de cette discipline d'interface, c'est bien entendu la question de sa pérennité, de sa stabilité. C'est là une discussion assez intéressante dans le domaine, à savoir si cette fusion entre disciplines va perdurer ou si chacune s'étant rechargée de concepts, de connaissances de l'autre, va repartir de son côté jusqu'à une reconvergence future, un peu comme une comète qui repasserait à proximité toutes les centaines d'années. On peut s'en inquiéter aujourd'hui, le retour d'un certain utilitarisme médico-technologique en recherche, nécessaire, je vous l'avoue, un tropisme retrouvé pour l'étude de l'embryon humain, pourrait recréer un schisme tel que celui créé il y a un siècle par l'émergence de l'embryologie expérimentale. C'est une question qui semble un peu théorique au premier abord, mais qui en fait reflète le conflit épistémologique qui existe entre ces deux disciplines historiques de la biologie, car en effet le développement et l'évolution ne sont pas régis par les mêmes principes. Le développement est une discipline qui est avant tout basée sur une hypothèse de récurrence. C'est parce que à chaque génération, on postule que l'embryon à venir ressemblera à l'embryon qu'étaient ses parents, que l'on peut faire des expériences et avoir des résultats. Il y a donc une possibilité d'une démonstration. Que s'est-il passé? Comment cela s'est-il passé? C'est une approche prédictive, puisqu'on a en ligne de mire un résultat à atteindre. L'évolution, à l'inverse, est une discipline basée sur une hypothèse de non-récurrence, puisque le premier postulat de l'évolution, c'est que rien ne se répète jamais. Donc il existe une possibilité d'explication. Que s'est-il passé? Probablement. Pourquoi? Mais on est toujours dans une approche rétrospective avec un résultat à expliquer. Et on voit donc que cette cohabitation est par nature instable, en particulier si l'on prend garde à respecter les fondamentaux de chaque domaine. Et ce n'est malheureusement pas toujours le cas, ce qui donne parfois à l'Evo-Devo un caractère peut-être un peu superficiel, qui souvent fait le bonheur des médias, tout en soulevant la vindicte des orthodoxes des deux bords qui doutent, à tort ou à raison, du succès des mariages scientifiques intercommunautaires. Je vous remercie de votre attention.
Merci pour ta très élégante lecture. Question. Peut-être je peux commencer. J'ai été troublé, Denis, plutôt au début de ton exposé. À un moment, tu as dit peut-être on est presque au bout des, enfin aux limites d'une certaine façon de l'évolution. J'espère que je.
Non, j'ai pas l'impression. Non, j'ai pas l'impression.
Par exemple, que les variables disponibles étaient.
Ah, bien sûr, oui.
Ouais, c'est dans ce sens-là, oui. Et je ne dis pas que l'homme c'est terminé après l'homme.
Oui, alors c'est.
Mais les variables disponibles pour l'évolution étaient moindres.
Voilà.
Est-ce que, par exemple, je ne sais pas, c'est une question un peu naïve, mais les éléments mobiles, transposons et influence sur le génome ne sont pas des éléments susceptibles de recréer des potentialités d'évolution?
Oui, alors les éléments transposables ont été très utiles par le passé pour introduire de nouvelles régulations, etc. Ce que je voulais dire, c'est que au fur et à mesure de la complexification des systèmes, la marge de manœuvre de ces systèmes se réduit, bien entendu. Donc on a une accumulation de contraintes. Et par conséquent, on passe à un système d'évolution très graduel, qui était le système du 19e siècle, en fait, première moitié du 20e siècle, à un système de variation qui est relativement plus saltatoire, finalement, qui avait proposé, alors, pas proposé Stephen Jay Gould, mais proposé des auteurs du début du XXe siècle, par lequel il y aurait moins de variations, mais de plus grande importance. C'est ça. Aujourd'hui, on peut se poser la question, quelle serait la prochaine étape, puisque on a déjà atteint un niveau de complexification tel que l'on peut difficilement imaginer quelle pourrait être une étape suivante. C'est ça que je voulais dire, c'est que ces systèmes, en fait, pour pouvoir prendre en compte le fait que le nombre de gènes n'a pas évolué, et pour autant d'évoluer des structures de façon spectaculaire, ont perdu la capacité d'évoluer facilement puisqu'ils ont dû complexifier leurs interactions. C'est ça, en fait, ce que je voulais dire.
Autre question? Non? Ah, pardon, je n'ai pas vu. Antoine.
Juste une petite question sur l'idée de la partie, les lois communes impliquant les génomes. Est-ce qu'on peut imaginer qu'au cours du développement, il y a un certain nombre de contraintes qui, en réalité, ne sont pas les mêmes que pour l'évolution? En gros, la question qui se pose est, est-ce qu'il y a une contrainte sur la taille des génomes? Ça change, suivant qu'on répond oui, il y a une contrainte, alors on comprend que les gènes soient limités en nombre, etc. Mais si au contraire on se pose la question et on se dit qu'il n'y a pas de contrainte, alors on a une propriété qui est très intéressante pour l'évolution, qui est que si les génomes en fait ont tendance à être longs, cela explique qu'on ramasse de l'ADN à l'extérieur. Et donc, à l'intérieur de l'organisme, on est une image de l'environnement. Donc, ça devrait être très différent pour la partie développement et la partie évolution. Qu'est-ce qu'on peut dire de ça?
Oui. Écoutez, il faudrait vraiment que j'y réfléchisse, mais je pense que si l'on regarde l'ensemble des tailles des génomes disponibles jusqu'à présent, je pense qu'il est difficile de faire une association entre une taille de génome et soit un type de développement. On est toujours surpris, récemment avec le génome de l'axolote ou du poisson osseux, du poisson à poumon, un génome de l'axolote qui est extrêmement grand. Alors on peut se dire l'axolote a des caractéristiques extrêmement curieuses, puisque c'est un animal néoténique. Donc, est-ce que l'on retrouverait cette taille de génome dans des animaux proches? Non, on ne les retrouve pas, en fait. Si on regarde dans d'autres amphibiens, chez d'autres urodèles, c'est pas autant. Donc, je pense que c'est très difficile pour l'instant de faire une association entre la taille du génome, son contenu en éléments transposables et le type d'animal. Mais je ne vais pas m'aventurer plus loin.
Ok. Écoutez, s'il n'y a pas d'autres questions, merci encore, Denis. Et donc maintenant, je donne la parole à Edith Heard, qui est professeur au Collège de France, dirige la chaire épigénétique et mémoire cellulaire. Elle a travaillé longtemps à l'Institut Curie et actuellement la directrice du laboratoire européen de biologie moléculaire à Heidelberg, qui est une grande spécialiste de l'épigénétique et entre autres de la façon dont le deuxième chromosome X des femmes est inactivé. C'est ce dont elle va nous parler puisque le titre de son exposé est « La vie avec deux chromosomes X ». Juste le temps que les diapos se préparent, j'en profite pour faire une remarque. Les trois conférenciennes cet après-midi ont deux points en commun. Un évident, c'est que ce sont des scientifiques hors pair, mais un second, c'est que ce sont des binationaux. Michael est américano-suisse, Denis Duboul est franco-suisse et Edith Heard est franco-britannique. Edith, à toi dès que c'est possible, j'espère.
et remercier Monsieur le Président, chers collègues. Et en particulier, je voulais féliciter Mike Hall. Je suis une grande fan. Dans les contextes européens, on a souvent croisé des chemins et je suis toujours très admiratif de la manière élégante qu'il arrive à faire non seulement sa recherche, mais aussi, je dirais, naviguer le monde politique européen. Donc, je suis ravie de savoir pourquoi, en fait, j'étais invitée. Parce que mon exposé sera quand même un peu différent des deux premières, mais je suis très honorée d'avoir été choisie parce que je suis franco-britannique. En fait, je suis gréco-britannique et française, peut-être. Donc, c'est pas grave. Voilà, enfin. So I will actually switch to English, though, which has now become my dominant language as I'm at the EMBL. In fact, as some of you here know, the dominant language at the EMBL is Emblish. I've tried to resist speaking Emblish, but I can just about still speak English. So yes, I'm going to tell you about life with two X chromosomes. It will be rather different to the previous two presentations. But I thought I would start by something that I think interests us all, and I'm not sure this is working. This is interesting. Okay. It's changed here, but ça ne marche pas du tout. Ah oui, il faut du temps. Oui, mais à ce rythme-là, je vais montrer 2 diapos, je pense. Moi, je vois ma deuxième diapo, mais vous ne le voyez pas. Bon. This is not going to be easy. Here we go. Maybe I should start changing all my slides now. That way, I'll get through them. So yes, before talking about life with two X chromosomes, I thought I'd talk about life with sex chromosomes. And we all know that sex is very important in evolution. And the way one determines sex, of course, can be multifold. It can be either environmentally determined or genetically determined. And my particular interest, of course, is in the genetic determination and the epigenetic variants one can find. And here I'm just illustrating some of the very dramatic differences that one can see between the sexes, what we call sexual dimorphism, where males and females can either be fairly similar, as you can see here in the case of us and our primate brothers and sisters and ancestors. But then on the other hand, there are some species where indeed sexual dimorphism is overtly very different. Some birds, you can see here where in this case the male is indeed rather flamboyant, the female is rather less interesting. And then in this case, you can see in some fish where the female is enormous and actually does most of the work, and the male is tiny and parasitic and just sort of hangs on to the female to do we know what. So there's a lot of dimorphism, and the mammalian species are the ones that, of course, I'm most interested in, where the sex chromosomes, 2 X's and 2 Y's. I'm sorry, this is really very difficult. Now there are things flashing up on the screen. So the females and males do manifestly vary in some cases. For example, as you can see here, in the case of calico cats, which are always female, they have 2 X chromosomes. Males, male brothers are either black or orange, whereas the females have these patches of orange and black fur. And that is due to an epigenetic process that allows the equalization of dosage between XX females and XY males, which is what I'll be telling you about in a minute. But before going there, I thought I would say something about what we now understand about the difference between the sexes, particularly in humans, and how, in fact, science, biomedical science, has largely ignored or been biased to sex and gender differences. And I really wish I could change my slides. Denis, how come you managed to get it to change? This is really bad. Okay, here we go. It's changed on the screen at least. Wait, je ne sais pas pourquoi c'est... Okay, I did actually, and it doesn't. Okay, now it's going a bit faster. Thank you. Yeah. I don't know. We'll find out in a minute. Thanks. So indeed, as I said, there has been a strong bias in biomedical science, both in research and medicine, that is to do with sex and gender. And I don't want to. I could spend hours talking about this, but in fact, we are building on just a few decades of research, really, in our understanding of how the sexes are different from a biological and from a, I would say, medical perspective. So there have been, many centuries of misconceptions. The medical field only took on females, let's say, in the last century or so. For many, many centuries and millennia, women were not even properly treated. I'm sure some of you have heard the hypothesis of the wandering womb, the uterus, which is the cause of all female health issues. This was something that the ancient Greeks and the ancient Egyptians came up with. Anything from a runny nose to obviously, mental incapacity, all of that was down to the uterus. So this has continued for centuries, and of course, both for religious and societal reasons, women were not often examined because it was a very male-dominated world. But we have caught up quite a lot, and now there's really starting to be a deep understanding that, in fact, males and females are much more different than even our appearances would show. In fact, there's even the concept that every one of our cells has a sex. And of course, these differences are both hormonal and genetic due to the sex chromosomes, but they're also linked to environmental factors and lifestyle differences. And in fact, here I'm just showing you an overview of the many different areas of, I would say, differential treatment that leads to sexual dimorphism. Of course, testosterone, estrogen, progesterone between the sexes. This is initially due to the sex chromosome differences and the fact that the Y chromosome encodes a protein called SRY, which is important for determining the formation of testes and male reproductive organs. But we're starting to understand that these hormonal differences and the metabolic differences that can accompany them are incredibly widespread and actually can vary immensely between individuals. The lifestyle factors I don't think I need to dwell on. We all know that there are differences in different societies, but in the kind of intake we have from our environment, and of course our microbiomes are different as well. That is the other dimension of sexual dimorphism. Our immune systems are incredibly different, and I'll be coming briefly back to that. And what really interests me and my lab is the genetic basis of this difference. The fact that females have 2 X's, males have 1 X and 1 Y. That is increasingly being understood to feed into the sexual dimorphism that can result in differential health issues as well. And in particular, the X and the Y, the mammalian X and Y, evolved from a pair of autosomes, of course, and gradually differentiated across evolution. So that right now, in humans, the Y has literally a handful of genes that are important. Some of them are incredibly important for male-specific features. I mentioned to you SRY. But there are many other genes on the Y that are also important, and this is only just starting really to be understood. Now on the X, there is over 1,000 genes, and as you'll see, the process that I work on and that my lab works on tries to understand how the 2 X's in females are differentially treated. And most of the genes on the X are expressed when there's one active X in both males and females, but most of them are silent. In females, there's one inactive X. Now, there are many genes that are Y-specific. There are some genes that are common between the X and the Y. These are in the pseudosomal region. And there are some genes, which I'll come to, that can somehow become expressed from the otherwise inactive X, and their dosage is critical. So going back to history, more recent history, people, even the tobacco industry, realized that there could be advantages to the XX status. This is really just a bit of a joke, although it was a real advertisement for Virginia Slims, which are especially for women because they're biologically superior to men. Now, if you actually read the information in this, you can see that, in fact, they weren't too far wrong. Women are actually different when it comes to, for example, developed sweat glands, color blindness, red-green type, day blindness, etc. So, you know, there are good reasons already that people were noticing these differences that were also genetic. But of course, then extrapolating that to needing to have a very specific type of cigarette, I think, was pushing it a little bit. But more seriously, we know now that there's a tremendous sex bias in disease. For many autoimmune diseases, the overwhelming majority of patients are actually female. Taking lupus as one example, where I think it's almost 90% of people with lupus are actually women. There are also a lot of differences in our susceptibility to infectious diseases. And cancer has much more of a male bias, especially the non-reproductive cancers are much more present in men. Now, why is this? Of course, there are hormonal differences, we know that, but there are also many, many differences at the level of gene expression that are due to the X and Y chromosomes. And I wanted to put this up here to make, one comment about medicine. And up until recently, most clinical trials were carried out only on men for, good reasons in some cases. One can evoke thalidomide, and of course, women in reproductive age should not be involved in such clinical trials. But the result is that, in fact, most medications that have been developed were typically just adjusted to the patient's size and weight, and so women are, in fact, being treated as little men, small men. And this isn't, obviously appropriate, but it's actually also a huge cost for drug companies because in the late 1990s in the US, 8 out of 10 of the drugs that were pulled off the market were due to the fact that they had side effects on women. So this truly is a problem that only recently has started to be understood. And I would like to preach for the fact that if we want to talk about personalized or precision medicine, one really does need to think about effective treatment for all individuals, male and female, and they do have to be treated differently, and they have to be treated differently also at different ages in order to be protected equally. So this brings me to life with 2X chromosomes, which is indeed the area that my lab is most interested in, and that we have been studying now for actually more than 2 decades. So as I mentioned, in mammals, 2X chromosomes are usually found in females, 1X and 1Y in males. Now that might not look like such a big difference on the surface, but it is actually a huge difference because of the number of genes on the X compared to the Y. And different organisms have evolved different strategies for compensating the dosage of their sex chromosomes so that, even though we do have differences, they really are not so dramatic, at least at one level. And yet there are some differences. But I just want to highlight the fact that, for example, if you look at the only really viable trisomy there is in humans, which is trisomy for chromosome 21, that leads to Down syndrome. And 21 is one of the smallest chromosomes in humans, and just one extra copy of that leads to Down syndrome. All other trisomies are developmental lethal. So having 2X chromosomes, and the X is one of the larger ones, compared to 1X, obviously does have an impact. And it was back in the 1960s that Mary Lyon, a geneticist, came up with the hypothesis that in order to equalize this dosage, one of the 2X chromosomes becomes inactivated repressed, silent during early development. And this choice is made and then it's memorized. So once one of the X's, either the paternal X or the maternal X, is silenced during development, that choice is then stably propagated. And this is where epigenetics comes in. And in fact, this epigenetic memory is one of the, I would say, paradigms in mammals when it comes to X inactivation. Now, of course, I mentioned to you that it could be either the paternal or maternal X, and this choice is made fairly randomly early on, which means that females are usually mosaics for the expression of either their paternal X or maternal X in each cell. And because the choice is made early, you end up with these clonal populations of cells that maintain the decision that was made earlier on to either express mother's X or father's X. And this is why you get these beautiful mosaic patterns of fur in calico cats, but also this is how Mary Lyon actually discovered the process looking at X-linked genes for coat color in the mouse. And in humans, we don't have fur, luckily or unluckily, but we do have other manifestations, and there are, for example, genes on the X that lead to disorders in sweat gland formation, and you can see the mosaicism here in a female who is heterozygous for such a gene or a mutation in such a gene. So basically, because of this random X inactivation, no two females, not even twins, are identical for X-linked gene expression, because although the two X chromosomes are largely the same, of course, there are a number of polymorphisms, differences. Every single gene has a little bit of a different sequence to it. So in fact, you get a lot of variation between individuals who are even though they can be genetically identical, because either one or the other X has been silenced, and the degree of mosaicism and its pattern can vary substantially. And now, this is a beautiful paper actually from the Nathan Lab, published now 10 years ago, where they were most interested, I think, in looking at the brain and neural development function. And they tagged a housekeeping gene, as we put it, a gene that's expressed in all cells, either with a green fluorescent protein or a red fluorescent protein. That way they could actually look at the expression in every single cell type within the mouse. And here you can see, indeed, that even between pups who are siblings and who should be identical, because in mice you can have very inbred mice, and so the X's can be pretty much identical, except for these fluorescent markers. You can see how different they are at this level. And when you zoom in into different tissues, for example, the skin, this is a section of the tongue, this is intestinal villi, you can see these patches of green or red cells, and these vary tremendously between individuals. And this is just one of my favorite images in this paper, where they've taken a brain and they've actually looked at a coronal section. And you can see that on one side of the brain, mainly mom's X is expressed, and on the other side, mainly dad's X is expressed. But in fact, this happens to be an extreme case. But you can imagine that this might have consequences in the way that neural function is achieved in females. And this is something that is of great interest in the field of neurobiology. So this X inactivation mosaicism that we see across tissues is due to many factors, the randomness early on, but then the ratio of founder cells to adult cells, the timing and the extent of cell migration during development, and also the alleles, because indeed, if you do have polymorphisms, they might be counter-selected early on in some cell types, so you end up with some tissues that are predominantly only expressing one X and not the other. So this is really a diversity-generating mechanism that can operate on all cells within a tissue and create diversity spatially that has the potential to add functionally relevant capabilities. So up until quite recently, we all thought that that was pretty much it, that, there's dosage compensation, one X is inactive, the other one is active, and this variation due to mosaicism is because of random X inactivation. But more recently, there's been an understanding that many genes on the X that is inactivated can actually escape this process. And it turns out that this can be an added layer of mosaicism, because although about 10% of the X-linked genes can escape in all tissues, and I'll show you in a minute, but these are the genes that tend to have a homologue on the Y, so there are two copies expressed, both in males and females. but about 15% of X-linked genes do this very variably between individuals. So you get a lot more variety in X-linked gene expression pattern due to this escape, which varies between tissues and between individuals. And there's an increasing understanding of where this variation might come from. Part of it is genetic, and part of it is environmentally controlled. There was a study that showed that indeed this kind of variation in identical twins is tissue-specific and associated with age, which means it must also be environmental. Now, I just want to say that across the X chromosome, we're realizing that there are many, many genes that can escape. More than 25% now, I think, is the estimate. Sorry, I don't seem to be able to control my slides. But here I'm just showing you, indeed, that the difference then between males and females is... I can't even touch it. Okay, maybe I can use a pointer. No, it's all right. It's fine. Ça va. J'espère. You can see that, indeed, there are many genes on the inactive X that are also expressed, and this leads to a double dose. And this question of dosage is becoming increasingly interesting and important for us. I already mentioned to you trisomy 21 and how dramatic that is. Well, having a double dose of some genes on the X can equally be extremely important, both for good or for bad. Now, many of these escapees I mentioned do not have a homologue on the Y. And these are often the ones that vary between tissues. As I said, some of this could actually be due to environmental influences. Some of it may be accidental. In other words, that there's instability at the epigenetic level of the silent state, and some of it could be purposeful. In other words, you need a double dose in an XX individual, in a female, because it's actually important for something. Some of this could be deleterious, and there's an increasing understanding that this escape from X inactivation may actually underlie several sex-biased metabolic, immune disorders, neurological disorders, and other diseases. And I mentioned lupus to you earlier. There's an understanding now that one of the genes on the X that escapes X inactivation that's involved in innate immunity, the toll-like receptor 7 gene, might contribute to lupus in this way. A double dose is actually correlated with the onset. Dear, oh dear, oh dear. Okay, this is not good. Okay, maybe you can carry on now? Yes. So I'm going to probably skip some slides because I realize that the time factor is kicking in. So I want to just tell you now some stories around the inactive X chromosome and how some of these genes can escape from the state. Now, just to step back, in fact, the inactive X chromosome, before it was hypothesized by Mary Lyon, had already been observed by cytogeneticists such as Barr. And in fact, for many of you medics, you know the inactive X as the Barr body, which was seen, in fact, originally in female cat neurons back in 1949 as a heteropycnotic body at the surface of the nucleus, often at the edge of the nucleus. Now, we've come a long way since then. Once X inactivation had been hypothesized, many scientists studied the genetic regulation of this process and how one of the two X's is chosen for silencing and then how it becomes silenced. Now, what I think is really important and fascinating in this area is that the two X's have to exist in the same nucleoplasm. They're in the genomes of the two X's, or the two chromosomes, are floating around in the same soup of factors, and yet one of them is on and the other one is more or less off. And the key regulator of this process, at least in eutherian mammals, is an RNA, a non-coding RNA known as exist, which is expressed early on in development from the chromosome that will become inactivated. This non-coding RNA coats the chromosome, decorates it in cis, and then triggers silencing. Now, exist is quite a large gene, and it encodes a protein, or it produces a protein of about 17 to 19,000 nucleotides, so it's a very long non-coding RNA. It's a fascinating molecule because it's a multitasking molecule. It actually brings in the factors that will silence the X chromosome, as well as some of the factors that will help to memorize the inactive state. And this is just showing you here the way the gene is organized and the RNA. And there are some regions of this RNA that are highly conserved. Most of it is poorly conserved. And the highly conserved regions are the ones that we think, or that we know, are involved in the process of exon activation. Now, once exon activation has been established, it's then maintained silently. It was believed for a long time over cell division. And this is a combination of epigenetic factors, including chromatin changes, reorganization of the 3D structure of the X chromosome, asynchronous replication. And there are several examples that I won't show you of molecular detail where we now understand how the inactive X is wrapped up and condensed. But I want to tell you actually more about how this happens. Now, I will not dive into details here. I want to just start by saying that many of us, for decades, were working on how do you switch a chromosome off. How do you switch all the genes on one X chromosome in an XX cell, and what does exist actually do? And here I'm just summarizing in one slide the fact that this non-coding RNA recruits a factor which hadn't previously been suspected to be involved in exon activation. It's present in both males and females, this protein, SPEN, but in fact exists very specifically recruits this factor. And what you can see here is just a cartoon. So this was in a paper that a very talented student of mine, François Dossin, we published a few years ago. SPEN is recruited by exist RNA, and it actually then brings in a set of complexes and factors that will then silence all active genes on the X. So exist will bring in SPEN. SPEN will then target genes that are on, and this will then lead to their silencing. And as soon as they get silenced, SPEN will disengage. Exist will remain, but SPEN will disengage. So it's a sort of hit and run process. Now that we thought was how the initiation begins, and we thought SPEN had its main role at this point. We thought that the maintenance of the inactive X was regulated by many other factors, also initially brought in by exist, including polycomb and other factors, as well as DNA methylation, which is a classic epigenetic mark. Now, my lab has become increasingly interested not only in the silencing, but in this escape. How do these genes break free from this, I would say, weapon of mass destruction that exists, is bringing in SPEN and its complexes to shut down genes? So we know that some genes actually escape right the way through development. From the beginning, they just never get shut down, whereas other genes get shut down and then they come back on again. Sometimes they come back on again in a very variable way, sometimes in a very specific way. So we differentiate these escapees, as we call them, as constitutive escapees and facultative escapees. And we've been interested in both types, but one thing I just wanted to highlight is that in vivo, some of the genes on the X that we knew were involved in various syndromes, such as, for example, MECP2 and Rett syndrome, it was believed that they were very stably silent, which is why, in fact, in females, when the mutation in MECP2 is on the inactive X, the active X produces wild-type MECP2, whereas on the other hand, if the mutation is on the active X, the cell is essentially nullisomic for MECP2. So there's been a lot of interest in finding ways to wake up MECP2 by waking up the inactive X chromosome for Rett syndrome therapies. And it was believed that this was actually almost mission impossible because of the very, very stably silent inactive state of MECP2. But what we had found was that actually during early postnatal development in the brain, there are some cells where MECP2 has come back on. So this is a whole area which I will not go into the details of, but this question of dosage and how you think that, you have a single dose of a protein encoded by, 1X, the active X, and in fact, in some cells, you can end up with a double dose. As I said, some of this could be actually helpful, and in other cases, it could be detrimental. Now, there's one gene on the inactive X, which I will not go into details of for the sake of time, but which escapes constitutively. It always escapes. So it's always expressed from the inactive X. And we showed a few years ago that, in fact, the double dose of this gene, UTX, is probably involved in helping to change the chromatin state of the inactive X chromosome during development. There's another gene on the X called ATRX, also involved in disease. And in this case, we think that this escape of ATRX happens very specifically in a cell lineage or in trophoblast giant cells, which is where, in fact, the protein itself accumulates with the inactive X. So you need a double dose, probably, of this protein, because it actually needs to become accumulated on the inactive X. So this suggests that maybe escape, indeed, has evolved in order to allow certain female-specific or XX-specific functions. Now, there are other situations where escape may be either helpful or problematic in diseases. And when I spent many years, happy years at the Curie Institute, we collaborated with medics there, and we were able to show that, in fact, the inactive X chromosome shows a high degree of epigenetic instability. Now, going back to Barr, who back in the 1940s originally detected the Barr body, he also published in 1957 the fact that the loss of the Barr body, so this is normal cervical tissue and this is abnormal cancerous tissue, the loss of the Barr body, the disappearance of the sex chromatin, as he put it, was a key indicator of aggressive tumors. So this was something that I think pathologists had been aware of for many, many years. And we decided to dive in and look at the molecular characteristics of this. And indeed, the inactive X is actually very differently folded and very differently active. There are more genes that can escape X inactivation in cancer cells, and this correlates with changes in the epigenetic state of the inactive X. So in this case, there could be an indication that this could contribute, in fact, to cancer appearance or progression. We were looking in breast cancer. But there are other cases, and this is a whole hypothesis that many people in the field are now exploring. It's called the exit hypothesis, which is that because of escape from X inactivation, some genes on the X, such as tumor suppressors, may be protective for females, which could explain why men have a much higher tendency of certain types of cancers, of many types of cancers, liver being one very, very dramatic example, but many others. And so it's not just hormonal, it could really be about X-linked gene expression. And I want to, actually I'm going to skip these 2 slides and show you a summary of this because, okay. So I just want to come now to the end of my presentation to say that we started to get an understanding of how this escape that can vary is actually controlled. And in fact, there have been some studies where this non-coding RNA that I told you was, the sort of weapon of mass destruction for silencing genes on the X. In fact, it was thought it only really mattered early on in development. But there's an increasing realization that this non-coding RNA is actually important for the silencing of X-linked genes, even in differentiated cells. And the degree of escape seems to correspond to exist. Now, there have been a number of studies, and the 2 slides I skipped were actually corresponding to this paper, which is a beautiful paper that was published by Christophe Ginestier in Marseille and Raphaël Margrand at the Curie Institute. And this was the work of a postdoc, Laia Ricard, where she showed that if you knock out or knock down exist in mammary stem cells, you actually inhibit their differentiation, and this can increase tumorigenicity. And what she showed was that this reduction in exist, either by knocking out the gene, deleting it, or reducing the levels, led to the increased expression of just a handful of genes, just 4 or 5 genes, one of which was a gene called mediator 14, MED14, which is a key factor involved in enhancer regulation and gene regulation, which you heard about earlier. And a little bit more of MED14 protein is sufficient to rewire the whole genome and block it in a stem cell state. So just a little bit more, or one factor, is sufficient to do this and may contribute to cancer. So this led us and many others to think about what could exist be doing. And I mean, there are some more recent examples. For example, in human B cells, it's been proposed that exist RNA may indeed prevent escape of X-linked genes. And so what we have done very recently is create a system where we can actually look at how much you can modulate escape. And this is the work of a wonderful postdoc who maybe some of you have heard, who's on the market to set up her own lab. I would say she's probably one of the most talented postdocs I've ever had, Agnès Eloda. And she stumbled across a finding, which is that if you can induce exist levels in a variable way using a trick, which is doxycycline sensitive promoter of exist on one X chromosome. If exist can come on, but you can actually increase the levels of exist artificially. And this then actually leads to, in an adult cell or in a somatic cell, increased levels of exist. And what Agnese and our collaborators, together with Antonia Houth, a student in the lab, were able to show is that this can lead to silencing of all SKPs, both facultative and constitutive. And the degree to which you get silence depends on the levels of exist and the time at which exist is expressed. So the longer you express exist at higher levels, the more likely you are to become completely silenced and irreversibly silenced. And at early stages, if you fluctuate the levels of exist, you can see these SKPs coming on and off. So I'm going to end here. This has, for us, many, many huge implications, because it actually means that we now have a tool, but also an indication of how these SKPs that might be very specific and very important for female physiology and health are regulated. So in summary, we think that this process of escape from exon activation is much more widespread and variable than ever before it was thought, both in humans and mice. Mouse is our favorite system, but we switch to humans when needed. And we're also intrigued by the different mechanisms by which genes can escape. I didn't say anything about that here, but we think there are specific factors that allow constitutive SKPs to stay on constantly. But all of these genes are sensitive to this long non-coding RNA exist. And in fact, now we realize that in human populations, the levels of exist RNA can vary, and they can vary through life. And this variation may indeed be absolutely critical to X-linked dosage variation, both in development and disease. And I will end there, and thank you for your attention and patience with the slides, and just acknowledge my wonderful lab who have bravely carried on as I have been EMBL DG, and fantastic collaborators, both past at the Curie Institute and present at EMBL. And thanks for your attention.
Merci, Edith. Great talk. Thank you. Questions? Yes, please.
Going back, thank you very much, Edith. Going back to the beginning of your presentation about the factor that women in general have not been fully appreciated in terms of their biology, would you say that evolution gives the fact that one of the species has two large chromosomes a certain advantage to the female? Because at the same time, you can look at birds where it's just the opposite. The male has two large Z chromosomes, yeah, and the female has a small chromosome. Do you see any philosophical things to discuss there? Maybe the birds have the males are more.
I think the only thing I can really say for sure is that there are so many different ways to determine sex. And the evolution of the sex chromosomes is hugely variable. Now, even amongst the mammals, eutherian mammals such as us have two X's or an X and a Y, and the X and the Y are actually very different. There are even some mammals, eutherian mammals, that have lost their Y. This is one of these big adventures. In fact, the Y is getting smaller and smaller, and there's some mammals, such as Elobius, a small vole somewhere in, I think, Ethiopia, has actually lost its Y chromosome. You still get males, and that's probably because a neo-Y has appeared elsewhere. But to go back to the differences, in monotremes, such as the duck-billed platypus, there you have 5 X's and 5 Y chromosomes. They're unusual creatures, but still, they do have clear sexual dimorphism. And then even amongst marsupials, where, again, there are 2 X's and 1 X and 1 Y, here, the dosage compensation, the degree of dosage compensation may be very different. The way it's regulated is very different. So I don't think we can ever say that there's an advantage or a disadvantage to having, 2 large sex chromosomes versus one large and one small. But clearly, having the presence of two sex chromosomes that can cover each other's back, so to speak, if there's a mutation, is an advantage. And that's why, you know, many of the X-linked diseases are well known, because of course in males, you only need one hit to see the disease, Duchenne muscular dystrophy, hemophilia. So yes, of course, there is some protection to having two copies of a large chromosome. But whether it's an advantage or not, I hope that what I showed you shows that sometimes it's not an advantage, because having a double dose can actually lead to severe problems for some X-linked genes. So, open questions, yes.
Oui, Mike. No, doesn't know. You have to.
I'll have to give your gold medal back soon if you carry on.
Not smart enough for this place. Yeah, very nice talking. I was wondering, in the case of XYY men,
yes,
you have two double Y. Is there similar Y inactivation in this case?
No, there isn't. Actually, that's a very good point. In fact, I'm right in the midst of writing a grant with a colleague, James Turner, who also works on sex chromosomes, and we're very interested in both the X and the Y in supernumerary copies. If you have multiple Ys, there is no hint of any silencing, but it is a problem, and it's been shown that extra copies of some Y-linked genes actually are very linked to certain types of cancers. And actually, being XXX is probably less problematic than being XYY, because in an XXX female, only one X will remain active, the other two will be inactivated. So it's only now, because of, you know, we have these huge biobanks, and for example, UK, Denmark, other countries, where you can start to really look at human tissues and different ploidies, and it's becoming obvious that there's a lot more sex chromosome aneuploidy than anyone ever imagined. And this goes back to a remark I made at the beginning, which that we think that even cells have a sex. And in some cell types, having an extra Y or not having an extra X or Y, being XO, can actually be problematic in a developmental context. And that can then lead to later problems in health. So this is, I think, a huge field that only now can be explored. No one ever suspected there was going to be so much diversity in ploidy. And many individuals are actually mosaic for their ploidy. Many XO females are actually mosaics of XX and XO cells, which is probably why they don't have as severe symptoms as if they only were XO, because most XO embryos die during early development.
Oui, Edouard Brezin.
Yeah, sorry for the naive question.
No, I like naive questions.
You said that one of the X which is inactivated is not controlled always by genetic processes, but by epigenetic other processes. In that case, why is it that when this gene is repeatedly active, why is it always the same X which is inactivated?
You see, that's why I like naive questions, because they're the killer questions. That is the $100 million question. We don't know. First, we don't know how exist, which is expressed from the otherwise inactive X, maintains its active state and doesn't forget to keep active as cells divide. So we still don't quite know how that works. It's probably an epigenetic process, but initially there was a choice made, and how do you keep that exist gene on? And then I think this is the more fascinating part of how do you set up cellular memory. Once you've expressed exist and you coat the chromosome, it's truly epigenetic in the sense that there have to be some changes that progressively become memorable and locked in and can replicate themselves. Because in fact, exist, every cell cycle, at least in the mouse, gets switched off during mitosis. It only starts to come back on after G1. So exist is not the pure memory factor. So there are many aspects to this. How do you keep the active exist allele on? How do you keep the rest of the X off? and what are the different factors that do the job. And just to make it even more complicated, we think that even between mammals, there are different ways to do this. Yeah.
Edith, une question à propos de la perte de mémoire chez les gamètes ou le zygote, je ne sais pas.
Oui, la reprogrammation.
Qu'est-ce qui se passe sur le X inactif?
Oui.
Comment est perdue l'inactivation?
Ça aussi, c'est une grande question. Nous savons que le X inactif est reprogrammé déjà, enfin, dans les gamètes, au cours de la gamétogenèse. Nous connaissons le timing, mais l'ordre exact des événements reste encore un peu inconnu et les mécanismes directs ne sont pas connus. Donc, clairement, existe et éteint, mais est-ce qu'il y a une reprogrammation active? Et donc, il y a une deuxième phase de reprogrammation que nous connaissons, que mon labo, entre autres, a découvert, c'est au cours de l'embryogenèse. Une fois que le X est inactivé chez la souris, c'est le X paternel au début, qu'au début, en fait, il y a une phase de reprogrammation dans la masse interne de l'embryon. Donc, c'est l'équivalent des cellules souches embryonnaires. Et là, nous avons réalisé qu'effectivement, pour cette reprogrammation, existe et éteint. Et puis, il y a une perte de cette mémoire progressive. Mais c'est là où on a trouvé, merci pour la question Alain, parce que c'est là où on a trouvé que UTX, ce gène qui échappe à l'inactivation, code en fait pour une déméthylase de l'histone H3K27 et probablement il faut une double dose de cette protéine pour faire ce boulot. Et on a trouvé qu'il y a une partie des gènes sur le X qui sont sensibles à cette déméthylase. Donc, on pense qu'il y a différentes manières de reprogrammer et réactiver. Et sans doute, la manière chez le gamète n'est pas la même que dans l'embryon. Donc, ça aussi, c'est des champs de recherche actifs. Voilà.
Françoise.
J'ai cru comprendre au début que tu disais que dans divers tissus, il n'y avait pas le même actif et inactif.
Oui.
Est-ce que c'est vrai? Et comment ça se fait que, par exemple, dans le sang qui va se propager dans les divers tissus, il n'y a pas un conflit ensuite?
En fait, une fois que le choix est fait au cours de l'embryogenèse, et c'est au stade, chez l'humain, on pense que c'est au stade où il y a quelques centaines de cellules, peut-être un peu moins. J'ai l'experte en face de moi, Claire Rougel, là. Chez la souris, c'est un peu différent parce que, bon, on sait que c'est, oui, quand il y a peut-être 120, 200 cellules, il y a une partie des cellules qui font ce choix. Et ensuite, ce choix est propagé stablement, ça c'est clair. Mais après, il y a d'autres facteurs qui vont influencer la migration des cellules. Et quand on a un allèle sur un chromosome X qui, par exemple, code pour une protéine où il y a un avantage de croissance cellulaire ou d'adhésion ou de migration, on va trouver qu'il y a certains tissus où il y a beaucoup plus de cellules qui expriment cet X-là. Et donc, par exemple, chez les femmes qui portent une mutation dans le gène qui est impliqué dans l'hémophilie, donc chez les garçons, tous les garçons sont malades quand il y a une mutation dans ce gène. Chez les femmes, en fait, souvent, elles sont complètement asymptomatiques. Et on pense que c'est parce que dans le sang, en fait, 100% des cellules n'expriment que le X actif avec l'allèle sauvage. Alors, pourquoi? Ça, c'est une question intéressante parce que apparemment, il n'y a pas de raison qu'il y aura une contre-sélection, au moins chez l'adulte. Donc, on ne sait pas à quel moment cette contre-sélection doit avoir lieu. Et maintenant, en fait, souvent, les médecins demandent, mais voilà, ils ont des patients qui présentent avec une maladie, mais quand ils regardent, en fait, et si c'est une maladie liée à l'X, ils voient qu'en fait, il y a beaucoup plus de diversité dans le mosaïcisme qu'ils imaginaient. Donc, c'est ça aussi, et c'est un champ très actif maintenant de recherche biomédicale. Ouais.
OK. S'il n'y a pas d'autres questions, merci beaucoup, Edith. Encore bravo et merci à Mike et merci à Denis et Edith qui nous ont entretenus, je pense, et nous ont inspirés. Donc, je vous propose maintenant d'aller boire un verre, un cocktail, et de discuter plus avant. Voilà.